Hello everyone. In this video we are going to learn the history of plant virology in brief. This video is going to be a part of a series of lectures to be delivered on the subject plant virology. Through the study of the history of the plant viruses, we will get to know the fascinating world of this intriguing pathogen. As we go on, we will know how the researchers have discovered and unraveled the nature of this submicroscopic pathogen. Though most of the developments in this subject took place in 20th century, the foundation of this subject was laid in the last decade of 19th century. However, there are a few reports of plant virus diseases much before that. Now let us see those early reports. The oldest record of a plant virus disease is seen in a brief poem written in the year 752 AD by a Japanese empress named Koken. It was about yellowing of eupatorium leaves. Now this yellowing is now believed to be vein yellowing disease, which is white fly transmitted, and this disease is more likely related to eupatorium yellow vein begomovirus. Now the second record of a plant virus disease is tulip color breaking, the famous tulip color breaking disease. It was first described by Carolus Clusius in 1576, only 16 years after the introduction of tulips into Western Europe from the Middle East. Now, this is transmitted by grafting of bulb. Now, what happens here due to this color breaking disease? Abnormality occurs in the flower, and we see stripes, longitudinal stripes in the flower, and the normal color of the flower is broken. However, due to this disease, the broken flower became more precious, and could fetch higher price than the normal flowers. And these led to the craze for having tulip color broken flowers, and this is popularly known as tulipomania, the great Dutch tulip trading, and it was so prized and traded that large sums of goods could be traded with one flower, color broken flower. So it was not known that time that this is due to a virus disease. Now the third case of early reports is the Abutilon mosaic, and it was seen in the countries of France and Belgium. Now this is also graft transmissible. Now let us move to the scientific study of plant viruses, the beginning of the subject. So all the examples that we have seen so far, those were there without any knowledge of virus in the mind of the people who witnessed the presence of virus diseases in plants. So before we really go into the scientific study or history of the subject plant virology, we should see here a technique that is called filtration technique, because this technique helped the researchers to know the real nature of viruses, to know that virus is something different from bacteria and fungi. So this technique is quite important to know. This technique was developed by Chamberlain, an associate of Louis Pasteur, It consisted of a candle-shaped filter of unglazed porcelain mounted on a suction flask. So through this filtration technique, bacteria and other microbes could be filtrated. That means when we have a solution, if that is filtered through this technique, then bacteria and fungi and other microbes could not pass through this filter. And the filtrate that was obtained was free from microorganisms. So with this technique that was there at that time during later part of 19th century was a valuable technique for virus research. So tobacco mosaic disease, the first virus disease to be described and studied. And who studied that? It is him, a Dutchman Adolf Mayer, in the year 1886. So he was practically the beginner of the study of plant viruses. He described a disease in tobacco and named it as mosaic disease. He showed that the causal agent of mosaic disease was transmissible by sap. So he was the first person to reveal the sap transmissible nature of the virus causing tobacco mosaic disease. And he could not find bacteria or fungi associated with the disease. However, he did not use the filtration technique, the Chamberlain filtration technique. He assumed that it could be a pathogenic bacterium, though he did not find any bacteria associated with the disease. He just found that a sap from the infected tobacco plant can cause disease in the healthy plant. He just noted that sap from an infected plant can transmit the disease to a healthy plant. So it was Adolf Mayer. After him, it was Ivanowsky, a Russian researcher, who took the study on tobacco mosaic virus forward, and he did the filtration. He used the filtration technique, and his observation was that tobacco mosaic virus was not retained by the filters. That tobacco mosaic virus was not retained by filters. His conclusion was that toxic substance secreted by bacteria could be there in the sap, or there may be small bacteria. He considered the agent as Contagium solutum. However, he also mentioned the occurrence of inclusion bodies in the infected cells. We will discuss later what are these inclusion bodies. Meanwhile, a rice grower in Japan, Hashimoto, he suspected the link between rice leafhopper, an insect, and rice dwarf disease. So it was the first known evidence of vector transmission of plant virus. It was also there in the 19th century, later part of the 19th century. Then came the last researcher in the 19th century who is now regarded as the father of plant virology. He was a Dutch microbiologist. He is Beijerinck. He repeated the filtration experiment. And after doing the filtration experiment, he suspected that the causal agent of tobacco mosaic disease is completely different from microorganisms. So this conclusion was phenomenal at that time. That indicated that there is something which is not bacteria and fungi but causing diseases. He did extensive dilution experiments. He also showed diffusion of infectious agent through agar medium. He named the agent as Contagium vivum fluidum. That means infectious living fluid. and he later gave the term virus, which means poison in Latin. After Beijerinck, there has been a gap of about 37 or 40 years, and there was no significant development in the study of plant viruses. Stanley, in the year 1935, crystallized tobacco mosaic disease agent by adding ammonium sulfate to the juice or sap collected from infected tobacco leaf. He also found that virus remained active after crystallization because the crystals he obtained by adding chemicals in the sap could produce mosaic disease when rubbed on healthy plant. He thought that it was only protein because he could crystallize it. His discovery in unraveling the nature of virus awarded him the Nobel Prize. Later, Bawden and Pirie they crystallized tomato bushy stunt virus and found that both tomato bushy stunt virus and tobacco mosaic virus contain phosphorus. And with this, they concluded that it is not a protein but it is nucleoprotein. So the agent causing tobacco mosaic disease or the agent causing tomato bushy stunt disease, they are nucleoproteins. That means there are nucleic acids and protein. So because of their discovery of the true nature of the agent, they were awarded Nobel Prize again. Next comes invention of electron microscope. Electron microscope was invented in 1930s by physicists. However, tobacco mosaic virus was the first plant virus to have been observed under electron microscope, and they measured the size of the virus as well, and it was found to be 300 nanometer in length and 15 nanometer in width. So it was a landmark development to see the virus for the first time. So now we know the nature of viruses, that is viruses are nucleoprotein, and we also know the shape and size of tobacco mosaic virus. Now so far the researchers have concentrated mostly on tobacco mosaic virus. So why tobacco mosaic virus? Why tobacco mosaic virus is the most studied and described plant virus? Because of its stability. It can be stored for 50 years at 4 degrees Celsius. And thermal inactivation point is 80 to 85 degrees Celsius. That means if the virus is heated at 80 to 85 degrees Celsius for 10 minutes, then the viruses will become inactive. So, tobacco mosaic virus has very high stability, and this virus is found in a relatively high concentration in sap, and transmission is also easy. It is sap transmissible. So these are the factors which helped tobacco mosaic virus to remain in the focus of research studies by the plant virologists. There are several firsts in virology associated with tobacco mosaic virus. So tobacco mosaic virus was shown to be consisting of RNA and protein for the first time. And tobacco mosaic virus was first characterized by X-ray crystallography. It was the first virus used for electron microscopy, solution electrophoresis, and analytical ultracentrifugation. Tobacco mosaic virus is also the first RNA virus genome completely sequenced. It is the first virus which was used as a source of virus gene used for developing transgenic. It is also the first virus against which plant resistance gene was characterized. And there were two Nobel Prizes awarded on the studies of tobacco mosaic virus, as we said earlier. Meanwhile, we have seen there are some good observations on the transmission of viruses by vectors. In the year 1939, Watson and Roberts they classified the nature of transmission of plant viruses by the vector into two groups. One is non-persistent and the other is persistent. Sylvester in 1958 introduced the term semi-persistent. Now these natures of transmission will be discussed later in detail. Williams and Wyckoff they gave a technique called metal shadowing technique. This technique enabled the researchers to study plant viruses in detail under electron microscope. So when we say electron microscope, it was transmission electron microscope that can be used to see plant viruses. It is not scanning electron microscope which was used in the first hand. So in the year 1939, the first photomicrograph of tobacco mosaic virus was taken in transmission electron microscope. Well, then Markham and Smith in the year 1949, they found two classes of particles in the purified turnip yellow mosaic virus preparation. So here we see a kind of virus which are spherical, and there were two types of particles in the purified virus preparation. One is light. The light ones had only protein and they are not infectious. And the second one is heavy ones. They contain protein plus nucleic acid which were infectious. So it throws some light on the role of each of the components of plant virus. RNA is infectious, protein is not infectious. So the disease causing component of plant virus is the nucleic acid or RNA. Myron Brakke in the year 1951 developed density gradient centrifugation technique. This technique helped the plant virologists to purify plant viruses from a crude sample. Hershey and Chase 1952, we all know that they established DNA as the genetic material. They experimented with bacteriophage. These are also viruses affecting bacteria and killing bacteria. And their observation was that DNA of the phage enters the host bacterial cell, not the protein. So they established DNA as the genetic material using virus as the study material. However, it is not the plant virus here, but this piece of finding has perhaps the greatest impact in the biology and the subsequent development of biological science. Morel and Martin in the year 1952 gave apical meristem culture for elimination of virus from infected plant. So as a plant gets infected by virus, the virus is usually systemic in the plant. That means it can be found, the virus can be found in all parts of the plant. Now, apical meristem usually remains free from virus because the rate of growth of apical meristem is higher than the rate of movement of virus in the plant. So the apical part of the meristem remains free from virus. So according to them, if that apical meristem is excised and grown in culture media, plant tissue culture media, then that apical meristem can be developed into callus, and from there numerous seedlings can be produced which will be virus free. So apical meristem culture can be used for elimination of virus from plant to get virus-free plant from an infected plant. Franklin 1955 proposed the full structure of tobacco mosaic virus, and then we have one landmark discovery with Conrad and Williams in the year 1955-56. What they have shown is that viruses can be reconstituted in vitro from the RNA and the protein components. That means, say we have a virus here with the solid circles as the proteins, and inside we have a rod like structure. Say this is your nucleic acid. So let us say that this nucleic acid is RNA and it is surrounded by protein molecules. So this is a virus particle. Here one virus particle, here we have another virus particle. So we have number of virus particles here. So these researchers, what they have said is these components, protein and nucleic acid components, can be separated. So RNA is separated here and protein is separated. And they found that this RNA alone is infectious. And when they are separated, they can be also reconstituted in vitro. So we get full virus particles, mature infectious virus particles again, and if these particles which are reconstituted from the components, when they are inoculated to plants, we get symptoms, and we extract the virus and we get the virus again. So complete infectious tobacco mosaic virus particles can be reconstituted in vitro from the RNA and protein components. RNA alone is infectious, and RNA can be transcapsidated in protein from closely related virus. So we'll see this in the next slide. So the detailed study of transcapsidation, what does it say? Say we have a virus A and its RNA is called RNA A, and the protein, we know that protein coat of the virus is technically called as capsid. Protein coat of the virus is technically called as capsid. Since virus is a nucleoprotein, so we have the nucleic acid inside and the protein coat outside, and this protein coat is called as capsid. Now we separate the components RNA and protein. We inoculate RNA into plants separately and protein into plants in separate plants. The plants inoculated with RNA will develop symptom. Plants inoculated with protein does not develop any symptom. So from the plants inoculated with RNA, full virus particles could be extracted. And from the plants inoculated with protein, no virus could be extracted. So this shows that these RNA molecules are enough to cause disease in the plant, and these RNA molecules, they multiplied in the plant, and then we get the full virus particles here. So where from this capsid comes? This capsid is a protein which is usually, this capsid is a protein which is synthesized from the genes which are there in the RNA. So RNA is the genetic material. Here it says that RNA is the genetic material. Okay, this is with virus A. Now say we have virus B. In the virus B, in the same manner, we have RNA B and protein B. And when RNA is inoculated to plant, we get symptom and we get full virus particles synthesized from the plant inoculated with RNA. And the plants inoculated with protein only did not show any symptom, and so no virus was extracted from those plants. That means proteins are not infectious, proteins are not causing disease, and proteins are also not carrying any information, genetic information of the virus, so no virus is isolated from the plant. Now, beauty of this study is here. Now say RNA of virus A RNA, and protein of virus B or capsid, okay, they are mixed together. So we get reconstituted virus. So protein of virus B has encapsulated the nucleic acid of virus A. So we get a kind of virus particles, and we separate them RNA. So we get RNA of virus A here, and we get protein of virus B here. Now we inoculate these RNA and proteins separately into plants. So the plants inoculated with the RNA molecules got symptoms, and we get the virus A. And the proteins when inoculated to plant gave no symptom and no virus. So here, though this virus is reconstituted, it has the nucleic acid of virus A and protein of virus B. The RNA, when inoculated to plants, those plants yielded only A type of virus. So it shows that RNA is the genetic material, and this RNA molecule has all the information, genetic information of the virus. So, and this RNA codes for its own code protein or capsid. So it got encapsulated by the protein of its own. It got the protein from the genetic information which is there in the RNA molecule. So this study shows that RNA is the genetic material in viruses. So this type of study is possible only with the closely related strains of viruses. So the viruses which are distantly related, say for example tobacco mosaic virus and then say cowpea mosaic virus, we cannot reconstitute the nucleic acid of tobacco mosaic virus and the protein of cowpea mosaic virus because they will be distantly related. However, if we take two strains of tobacco mosaic virus, then this kind of study will be possible. Next is a separate study by a different group of researchers. They also found that it is only nucleic acid of tobacco mosaic virus which is infective. Then Crick and Watson in the year 1956, they observed that tobacco mosaic virus particles are composed of one nucleic acid and many identical protein subunits. And RNA does not have the coding capacity to make many different subunits. That was their observation. Meanwhile, we have some other developments like Brandes 1957. He gave leaf deep method for electron microscopy. Leaf deep method is a quick method to see viruses under electron microscope. Then another important observation was by Bancroft and Caseberg. They observed that alpha-alpha mosaic virus contains multiple components. There are many particles constituting one virus. That was their observation. Then we have some very significant discoveries in this time. Hewitt et al. In the year 1958 first showed the nematode transmission of plant viruses. Then subsequently we have discovery of fungi as the vector of plant viruses. Then discovery of satellite virus in the year 1962 by Casanis. Satellite viruses are the viruses which depend on other virus for replication. So the first satellite virus was observed associated with tobacco necrosis virus or TNV. So here TNV or tobacco necrosis virus is a helper virus, and one virus which is called satellite tobacco necrosis virus is dependent on tobacco necrosis virus. So it is not a separate name for the satellite virus since it depends on another virus, helper virus for replication. Its name is based on the helper virus only. So the helper virus is TNV and the satellite virus is called satellite TNV. Then Black and Markham in the year 1963 observed that wound tumor virus has double-stranded RNA as genome. Shepard et al. 1968 found cauliflower mosaic virus having double-stranded DNA as the genome. So plant viruses have single-stranded RNA as genome, double-stranded RNA as genome, single-stranded DNA as genome, and also double-stranded DNA as genome. Brandes and Waiter 1959 classified plant viruses based on morphology and serological affinities. Casper and Klug in the year 1962 gave the structure of tomato bushy stunt virus when they were studying the virus using X-ray crystallography. So, tomato bushy stunt virus was the first icosahedral virus for which structure was determined by X-ray crystallography. Otherwise, we know already that tobacco mosaic virus is the first plant virus whose structure was solved by X-ray crystallography. Then International Committee on Nomenclature of Virus was established in the year 1966 and it was renamed as International Committee on Taxonomy of Viruses in 1973. This is the apex body of virus taxonomy. They gave their first report in the year 1971 and the 10th report was published in the year 2017. So all the taxonomic problems are solved by this international committee. Now they have given several reports and in all their reports we see the updates on plant virus nomenclature, identification and classification. Then one landmark development happened in the year 1986 when the first transgenic plant was developed. It was transgenic tomato plant and this transgenic tomato plant was developed to show resistance to tobacco mosaic virus. And this resistance in the tomato plant was obtained by taking a gene from the virus itself, tobacco mosaic virus itself, and that gene was the coat protein gene of the virus. So this is also an example of first pathogen mediated or virus derived resistance in the plant. So this is the first transgenic plant ever developed and this is against a plant virus using a virus gene as the source of resistance. Then in 1990s transgenic papaya was developed. Papaya varieties like UH rainbow, UH sun UP resistant to papaya ring spot virus were developed using the coat protein gene of the virus itself. And these were the first commercialized transgenic varieties of plants. Then we have a long list of scientists and their contributions, and all these discoveries or inventions helped in the research of plant virology and also other branches of science. For example, in the year 1923, aphid was discovered as a vector of plant viruses. Then attenuation of sugar beet curly top virus was seen. TMV infected plant have antigenic property. Then local lesion assay, phenomenon of cross protection, use of indicator plants in virus studies, invention of electron microscope, then serological differences among viruses, then an observation that Asteriolus multiplies in its leaf upper vector. Now we have a great deal of efforts in plant virus nomenclature. The first attempt to name a plant virus was made in the year 1927. Of course, we know that Adolf Mayer first gave the name mosaic in tobacco, mosaic disease in tobacco in the year 1886. James Johnson in the year 1927 opined that plant virus should be named after common name of the host in which it was initially found and be given a distinguishing number. So the first virus seen in tobacco should be called as tobacco virus 1. Second virus should be named as tobacco virus 2 and so on. It was the first proposal to name plant viruses. Smith in the year 1931 introduced the letter codes X, Y, A, S, etc. for a number of potato viruses, and these letter codes are still used in naming some potato viruses. We still use potato virus X, potato virus Y, potato virus A, and so on. Smith 1937, he modified Johnson's system by replacing the common name of the host by its Latin generic name. So the tobacco virus 1 was called as Nicotiana virus 1. So it is another attempt to name plant viruses. So the efforts continue. Then Holmes in 1939 named genera of viruses on the basis of symptoms, while the name of the host plant was used for virus species name. So tobacco mosaic virus became Marmor tabaci. It is a symptom Marmor, that is mosaic, and tabaci is the host. So this is the first attempt to give scientific name of plant viruses in line with the Latin binomial of other organisms like plant, animals, insects. However, this proposal was not accepted. Then Hansen 1956 introduced more descriptive names. Potato virus X was named as Minflexus solani. So it is also a scientific name. Genus species concept is here, but the genus name has many codes and meanings. M denotes in Minflexus, in the genus name Minflexus, M denotes mechanical transmission, IN denotes virus without specific vector, and flexus denotes particle type. It was the first attempt to name and classify plant virus based on their properties. So a number of property of the virus can be seen in the genus name, but this became very complex name again. Gibbs et al. 1966 suggested that the vernacular names or common names should be supplemented with cryptograms composed of coded data on some properties of the virus. Now these cryptograms were given, this became more complex to remember. So after all these attempts, vernacular or common names were retained by the international body, that is ICTV, and tobacco mosaic virus name was retained as tobacco mosaic virus. Tomato bushy stunt virus name was retained as tomato bushy stunt virus. So these are the vernacular names which are composed of the name of the host and the symptom they produce. in the host, and these names are internationally accepted. Then we have a series of developments in the years 1970 to till date. Harrison et al. In the year 1971 classified plant viruses into 16 groups, and he also coined the term Geminivirus. Diner in the year 1971 discovered potato spindle tuber viroid. So viroid will be another entity or agent of plant diseases. These are different from viruses, and he coined the term viroid. Well, he also remarked that Altenburg in the year 1946 coined the term viroid. Then Kohler and Milstein invented the hybridoma technology for monoclonal antibody production. ELISA technique was discovered. Then this ELISA technique was used for plant virus detection in the year 1977. Then H. Sanger first sequenced the genome of potato spindle tuber viroid. Viroids are nothing but small single-stranded naked RNA molecule. So first potato spindle tuber viroid complete sequencing was done by H. Sanger. So it was the first pathogen sequence to be determined in the year 1978. Frank et al. They did the genome sequencing of double-stranded DNA of cauliflower mosaic virus. It was the first plant virus genome to be sequenced. Then nucleotide sequences of tobacco mosaic virus was done, though we see that tobacco mosaic virus is the first studied and described plant virus, but its genome sequencing was done after another virus, double-stranded DNA virus, that is cauliflower mosaic virus. Then virusoids were discovered. Prions are discovered. Prions are nothing but infectious proteins which are known to cause diseases in animals only. Then one very important discovery was made by Paul. He first transcribed the infectious viral RNA in vitro from the cDNA clones. So basically RNA to DNA formation, and then again DNA to RNA formation. So forming DNA from RNA is reverse transcription. So viral RNA reverse transcribed to DNA, and then these DNA, cDNA clones were used to make viral RNA molecules. So in vitro produced RNA molecules are also infectious. His study brought the reverse genetics to RNA viruses. Then many other observations like antibodies can be produced from transgenic tobacco. Then commercialization of cross protection, though it was not successful in large scale and it is not feasible in large scale. There are many risks involved in cross protection. Then RNA is critical component in resistance, in the pathogen mediated resistance. Discovery of proteins in the aphid as receptor to play a role in virus transmission. Then getting the Nobel Prize for RNAi or RNA interference work. So these are some major developments that happened in the subject plant virology. Each and every progress in the study of plant virus has a lot of influence in the subsequent studies. So big or small, each observation noted with scientific and logical conclusions bring about lots of new avenues for the future researchers. Even now, there are hundreds of labs all over the world working on plant viruses and discovering new things. So, studying the history of a subject is never ending. So, you should try to keep updated with the recent developments in the subject. And this lecture gives only some important discoveries or inventions that led to the establishment of the subject plant virology. So for further readings you can refer to these references. Thank you very much.